Welcome to another one of our interviews. I'm John Aguirre from the University of York here in England. Joining us today from Madrid is fellow main editor Mark van Rijk. Hey Mark, how are you doing?
Hello, John. Yes, happy to be here again.
I'm also very pleased to introduce Kushol Gupta of the University of Pennsylvania. Kushol edited the paper we're discussing today. Hey Kushol.
Yeah, good morning, John. Good seeing you all.
Great to have you here. Today, we're interviewing the authors of a really interesting review with the title Preparing for Successful Protein Crystallization Experiment. I'm sure you've noticed that we have some lovely crystals on the cover of our July issue, right? So you're going to meet the people who wrote about them, beginning with our first author, Gabby.
Hi, everyone. Great to see you guys this morning. My name is Gabby Budaszewski, and I am the operations manager at the National Crystallization Center at UBHWI. And I was lucky enough to actually attend this workshop that this paper was sort of written about, the SAMPREP workshop at ACA 2023. And Sarah and Vivian invited me to contribute to this paper to share some of the tips and tricks I learned over the years to prepare great samples for biomolecular crystallization and to kind of provide some context for the details of a crystallization experiment.
Wonderful, thank you. Next, we have Vivian.
Hi, good morning and good afternoon to Mark and John. I am a scientist at the Center for Biomolecular Structure at NSLS2, and one of my hobbies is to play with crystallization using different kind of off-the-track methods. So I did contribute, and I did help a little bit writing the paper.
Thank you very much, Vivian. And finally, our last author, Sarah.
Hi everybody. So my name is Sarah Bowman, and I am an associate professor in the Department of Biochemistry at Jacobs School of Medicine and Biomedical Sciences at University of Buffalo, and I'm also the director of the National Crystallization Center here at UBHWI. And so I was really excited to work with Kushol and Vivian and others in actually organizing and developing the SAMPREP workshop. And it was great to actually also then put together this manuscript that actually tried to capture some of the tips and tricks for crystallization. So I'm excited to be here. Thank you for the invitation.
Wonderful, great to have you here. Well, related to what you've just said, I think the first question will come from Kushol.
So, Sarah, just to continue on with what you just said, how does the review, do you feel, relate to the broader goals and the motivations of that ACA SAMPREP 2023 workshop that you just mentioned? I remember fondly we had originally met in 2022 at the ACA meeting to kind of discuss the original ideas behind this effort.
Yeah, so a group of us met in 2022 and discussed how it would be really great to actually put together a workshop that combined a lot of best practices in terms of sample preparation for different structural methods. And so, the workshop itself covered, I'd say, a wide breadth of different techniques, from sample purification to biophysical characterization to setting up Cryo-EM and crystallization samples. And we even had a session on how to ship, the best ways to ship your kind of precious biological samples. And so, I think the workshop overall was very successful. We had really great presentations, and I think we had really engaging discussion with our participants. And so our goal with this paper was to really provide a current best practices for sample preparation for crystallization experiments. And, we're really just excited to be a part of this special issue that Kushol is the guest editor on. So thank you.
Yes, my turn to ask a question to Vivian. One of the things you mentioned in the review is that AlphaFold may be used to identify and then remove flexible regions of proteins or nucleic acids before crystallization. Do you have any specific case studies where this was helpful?
No, I don't. I really see AlphaFold, especially AlphaFold 3, as one more tool to be used in crystallizing our samples, perhaps helping us with exploring the phase diagram, exploring how we could improve ligands or substrates or metabolites to actually bind to the molecule. Pretty much, I envision it more as material scientists have been developing new compounds by really trying to bring together different atoms. In our case, it would be we are bringing together different molecules. I can see that really removing certain flexible areas that we could improve the crystallization and we could actually improve the structure. But really probably at the moment, the strength of these AI methods is to help with this structure solution and serving as starting models for replacing in a way the phasing process of molecules.
My next question is to Gabby. Table 1 on the paper, it provides half-lives for DTT, BME, and TSEP. How do you balance reductant lifetime versus the side effects over time when crystals take, an eternity to grow?
Right. Yeah, great question. So I'll kind of bring it back to the reason why we need to have reducing agents around in our buffers at all. And, the reason why is because we want to keep our disulfide bonds reduced, especially because it can often lead to aggregation, which interferes with the crystallization process. And so if your protein takes a super long time to crystallize, I think I would probably recommend TSEP because it has the longest half-life and kind of the broadest pH range. So it's just a good general reducing agent. It is kind of on the more expensive side, but it's worth it if you have, a sample that you want to crystallize of your very precious protein. If you need an even more long-term solution than TSEP can provide, you could consider performing your experiments anaerobically, which can limit the amount of oxidation your sample undergoes. But I also kind of urge people to make sure they're also looking into the kind of unintended interactions that you might have between your reducing agent and other parts of your sample. So for example, metal compounds and salts will sometimes interact with beta-mercaptoethanol, especially like cobalt and copper. So that's going to affect its lifetime in the sample. So overall, I just urge people to kind of take a moment to very carefully consider your sample buffer because it can be super important for improving the results of your crystallization experiments and even like assay-based enzymology and all sorts of things that you're trying to do with your protein of interest.
Thank you very much. Those were some really great tips.
I think I have the next question here for Vivian. So Vivian, you discuss this really cool concept of micro-batch crystallization under applied electric fields. And you mentioned the lack of commercial devices. How close are we to seeing scalable, standardized systems for electric and electromagnetic field-driven crystallization, do you think?
Not too close, unfortunately. I think the application of electric fields and magnetic fields or electromagnetic fields, so to say, came from the fact that people have observed improved crystallization structures in the lack of or very low gravity. So it's the aim to improve your crystal quality by using fields. And that is then, so several groups have been playing with electric fields, not so many with magnetic fields. And so the idea of using the microbatch under oil developed by Naomi Cheyenne in combination with electric field really resulted in this device where the microbatch plate becomes the medium between two leads. So basically the sandwich capacitor. So, and really see how much the electric field affects the quality of these crystals. They do, but it's probably different for different proteins and different macromolecules because the electrical structure of the molecules, as we know, is different. So one of the limitations of the device that's being shown in the paper is that it has very fixed voltages, which were suggested by Professor Abel Moreno from UNAM. He observed that these different 1, 2, and 4, and 6 kilovolts was something that affected some of the commercial proteins. Regarding magnetic fields, I have been playing with low magnetic fields, not so low, but lower than what is usually reported, which are taking advantage of NMR facilities and actually grow the crystals in NMR tubes. And you also see the real nice alignment of the crystals in these tubes. Again, commercially, we don't have. It's still a lot of experimentation. But foremost, we need really to understand the interaction of the macromolecule with electric fields and magnetic fields, which is something that is not really very developed in the field.
Thank you, Vivian. That's very interesting. I have a question for Gabi on another technique to improve crystallization. In the review, you mentioned matrix microseeding and also cross-seeding. In your experience, how frequently does cross-seeding work? Or is there any correlation with how homologous the target protein is to the protein sequence of the seeds?
Great question. So personally, I don't have too much experience using cross seeds. I would say primarily I would do a cross-seeding experiment with like an apoprotein to try and nucleate crystals of a protein with a ligand. But there's a lot of information in the literature about these sorts of approaches. And so when I saw that you were going to ask this question, I kind of dug a little bit further into it. And I found this really neat paper about cross-seeding with antibody fragments by Obmalova et al. And they show that cross-seeding with different fabs can help to nucleate crystals that are often even better than just growing the de novo crystals. So in this case, it's, you know, something relatively homologous, but I also found another study by Abu Hamed et al. In 2016 showing that cross-seeding with proteins of low sequence similarity, but a relatively similar fold, so about like 20 to 30% sequence similarity, can help to nucleate crystals. And then there was another work by Caspi et al. Earlier this year that uses crystals from completely unrelated proteins. So seed crystals from completely unrelated proteins, just a mix of these to nucleate crystals. So it's sort of seeming like you don't even necessarily have to have any homology at all to use these sorts of seed crystals. And people have even used things like synthetic seeds, like functionalized nanoparticles, for the seeding experiment. And if you think about it, it sort of makes a lot of sense because I think a lot of us have seen results in our crystallization experiments where things like hairs or fibers actually act as nucleants for crystals. So it seems like there's a lot of potential to explore what sorts of seeds can be used to generate protein crystals.
Very interesting. Thank you.
I have a question for Sarah. With advances in serial crystallography, how have your sample prep recommendations evolved for micro and nano crystals? And can standard crystallization screens be adapted easily for generating usable microcrystals?
So that's a great question. So my lab actually does a lot of work in the area of nano and microcrystallization. And so some of the things that we find is that really for kind of successful preparation of these really ultra small crystals, you really still need to have kind of pure homogeneously distributed samples. So kind of all of the purification and biophysical characterization steps that we really covered during the SAMPREP workshop are still really important for making these crystals. And in terms of kind of the crystallization screens, there are definitely times when we see crystals that are super small that maybe in previous times might have been considered failures because really the crystals look like precipitate. And I think it's pretty exciting because these days we can actually use these samples and we can use them for techniques like microfocus beam lines at synchrotrons or X-ray free electron lasers and electron diffraction methods. All of these techniques really make these really small crystals suddenly usable. So all of that being said, when our kind of goal is to generate ultra small crystals, we really think a lot about the phase diagram and how to kind of drive micro and nano crystal formation. And so we end up using a lot of seeding techniques to generate really consistently sized micro and nano crystals. And honestly, we're really actively working on developing kind of methods that do make it more adaptable for people to kind of really easily make these kind of crystals in this size regime. So
that's great to know. Thank you.
yeah, thanks.
Okay, and I have one last question for Sarah. In what scenarios do you recommend pursuing crystallography first versus cryo-EM or computational predictions?
Yeah, okay, another great question. So, I think that there's definitely times that crystallography will end up providing kind of maybe faster and higher resolution structural results than other methods at this point. One of the things I think about is like the pandemic. This was really actually clearly demonstrated in the pandemic because so many of us really shifted to studying the proteins from the SARS-CoV-2 virus. And so, one of the things we did was like an analysis examining structures deposited to the PDB. And we found that if you look at the molecular weight and resolution, most of the larger molecular weight protein samples, those structures were mostly solved using Cryo-EM. And with some of the smaller molecular weight samples, those were mostly solved using crystallography. And so I think, for us, that really kind of helped us say, okay, both of these techniques are really critical for being able to kind of span the entire molecular weight scale. So, in terms of when you would first use Cryo-EM, if you're looking at something where you have a very large structure or a complex, I might try thinking about Cryo-EM before crystallography. In terms of kind of what advantages crystallography might get you, again, potential inhibitors, substrate binding, metabolite binding, things like that, we really see that that is still, I would say, dominated by kind of using crystallography methods. And I think that computational predictions are great tools to help us like design protein constructs and start to see some of the details of kind of different molecules binding. It's been my experience that to get to kind of the atomic level resolution, to actually see kind of things binding in an active site, that typically crystallography is really still the technique that will give us the most information. And so, you know, I also just want to say that it's a really amazing time to kind of be a structural biologist. You know, we've got this explosion of amazing computational tools. And then combining that with the kind of really just new capabilities of things like Cryo-EM. And what this is all enabling us to do in kind of an integrative structural biology approach is to kind of work on problems that are more difficult. So we can now kind of do things like time resolve studies to examine details of molecular mechanisms. You know, we're really well poised to kind of probe at physiological details. And I think it's just a really exciting time to kind of have all of these amazing new tools available to us. So.
Thank you.
I must say, I completely agree with your answer. It's a really exciting time. We've got so many tools at our disposal these days.
Yeah.
Right. We've come to the end of the interview. We've run out of questions. I would like to thank the authors and also Kushol today for joining us. Thank you, everyone.
Thank you. Thank you for having us.
Thank you.